Hi everybody, Dr. Biology here. This video is about evidence for mass flow. So if you haven't already seen my translocation mass flow hypothesis video, please do check it out right now. So firstly, let's look at the spec. So if we look at the spec, it does talk about that you will need to know about the use of what we call tracers and ringing experiments to investigate transport in plants. And then you need to recognize correlations, causal relationships, interpret evidence from tracer and ringer experiments to evaluate. So look at the pros and cons of the evidence for and against the mass flow hypothesis. So let's look at that. So I'm going to go through some exam questions as well to help you. So the first thing, let's look through the stem. So if we cut the stem, we can actually see the vascular bundles and the phloem are situated on the outside of the stem and the xylem on the inside. And that's important for certain experiments that we're going to discuss. So the first kind of experimental evidence that I'm going to talk about is what we call ringing experiments. So you can see on the diagram that they've cut a bark is removed. When they remove the bark, they're removing the phloem, but not the xylem. So what they then find after a while, there is a bulge or swollen region above the ring, and this fluid has higher concentrations of sugars than the fluid below the ring. And this is because the sugars can't move past the area where bark has been removed. Why is this supporting evidence? Well, this evidence shows that there can be a downward flow of sugars from source to sink. Another kind of experimental evidence is using aphids. So aphids have stylets that can actually reach down into the phloem. And what we can actually do is access tree sap via this severing of the aphid stylet and then actually analysing that sap. And the sap flows out quicker near the leaves than further down the stem. And this is evidence that there is a pressure gradient. So there's a higher pressure at the source and a lower pressure at the sink. So that again is supporting evidence. Next one is radioactive tracers. So this is quite interesting, radioactive tracers. So they use radioactive isotopes, the tracer of carbon dioxide, because that's taken up in photosynthesis and therefore it incorporates into glucose and then sucrose. The plant is then frozen to fix it quickly after it's been growing, and it's placed onto a photographic film in the dark, and evidence shows that the sugars are transported down from the leaf to the roots from source to sink, as you can see in this diagram. What evidence though is there against mass flow? Well, there's two main areas. The fact is that sugar does travel to many different sinks, so it doesn't just travel down the plant, it could travel up the plant as well. So it's and also not just the one with the highest water potential, as the model would suggest. Secondly, if you remember back to the structure of the phloem in my translocation video, sieve plates would create, and they do create, a barrier to mass flow. So you need a lot of pressure would be needed for the solutes to get through at a reasonable flow rate. Those are the two main areas of evidence against mass flow. Right, let's look at some questions. Okay, so I'm just going to talk about this question so you can see that it says here they investigated the movement of organic substances in 4 plants, A, B, C and D. 1 leaf from each plant was supplied carbon dioxide containing 14C. Figure 1 shows one of these plants. Each plant was treated differently before it's supplied with a radioactive isotope. A was ringed at position X, so we know what ringed means, so that they're removing the phloem. B at position Y. C at X and Y, and D wasn't ringed at all. Okay, so they ask you some questions there. I'd like you please to pause and we will go through the answers in a second. So A, all 4 plants were kept in bright light for 1 hour. Why would that be the case? Well, basically to allow rapid photosynthesis to produce those sugars that are going to be radioactively labelled. B, figure 2 shows the distribution of 14C after 1 hour. Which plant is shown in figure 2? Well, for 3 marks, it is in fact plant A. The reason being that there's no 14C at the top of the plant, and basically 14C is in the roots. Okay, so no 14C at the top of the plant, so it can't be X. 14C in the roots, so therefore it can't be Y, and therefore it must be A. C, use the information, explain the purpose of including plant D. So that's obviously the control. And why is it the control? Well, it allows you to compare distribution with an unringed plant where the phloem is present. Here's another one. Okay, so very, very similar in this case. So it says 1 leaf on a young plant was supplied with carbon dioxide. The plant was kept in bright light for 1 hour. The amount of radioactivity was then measured at 3 places in the plant. So you can see at the root, the stem and the bud. It says the diagram shows the results. Only the treated leaf is shown. Okay, so I'd like you to again pause and have a look at those questions. So, suggest one explanation for the difference in the amount of radioactivity in the bud and the roots. Well, you could argue that there's more carbohydrate transported to the bud than the root because it's growing. Carbohydrates are needed for growth, so that's why the bud would grow, and it's also needed for respiration, to release energy, and you could also synthesize as new molecules. So any of those points for 2 marks. Suggest why some radioactivity remains in the leaf. Well, it's going to remain in the organic molecules and it won't be therefore removed. So there's not enough time for it to be removed in 1 hour. It will eventually degrade, but it will take some time. Describe how ringing experiments could be carried out to determine which tissue transports the substances containing the radioactive carbon. So you could talk about removing the phloem. You can mention a named technique. So method for measuring radioactivity. I didn't mention that. You could use radiography. You could analyse feeding aphids, as we just said. If no radioactivity passes the ring, then transport is via the phloem. Okay, on to an actual exam question here. So I'm only showing you part of this exam question because this is an evaluation question. So this question is quite interesting because it shows different kind of experimental procedures, and it says investigating the effect of substance X on the growth of new roots. So it's kind of about the phloem, but it's also about growth as well. So it tells you that they use a ringing experiment to investigate the movement of substance X in stems taken from lemon plants. So cut out a length of stems, you then put a small block of agar. So remember, agar on top of each length of stem. They grew the length of stems in the same environmental conditions for 6 weeks, and then they found the number of root lengths of these per length of stem, so roots per length of stem. Roots grew at the other end of the stem from where the agar blocks place, so at the bottom. Okay, the table below shows the scientists' results. So the question asks about the mass flow hypothesis. It's used to explain the movement of substances through the phloem. Evaluate whether the information from this investigation supports the mass flow hypothesis. Do not consider statistical analysis in the answer. So I'm going to pause it and I'd like you to have a look at the answers. So in support of mass flow, you can see that with F, when the phloem is removed, there's very few roots per length of stem. So it is saying that the phloem is involved. Second point, so you basically need 2 of these points, by the way. You need 3 either support or 3 against, or 2 and 2. I'd probably do 2 and 2. So the second one, G, if you look at G, it shows that respiration and active transport is involved in the flow or movement. That's because at 4 degrees Celsius it slows or stops the flow. So as you can see, the mean number of roots per length of stem. So it's even though the phloem's present, there's not much active transport or respiration going on. 4, the agar block is the source, as you can see, and obviously roots are the sinks, because it tells you that the roots are growing at the other end of the stem. So source and sink, that's in support of mass flow. Why is it against the mass flow? Well, if you look at F, you'd expect the top part of the stem to have, above the ringing, to have a bulge. Now, there is no bulge. It's probably, well, it was for 6 weeks. You'd expect there to be a bulge in the stem. It could say there's no role for osmosis. So obviously osmosis, hydrostatic pressure, water movement. Water's going to be really important for the growth of roots. Movement could be due to gravity. So basically moving down via gravity, diffusing through. Roots still grow as well without the intact functioning phloem. I think that's the killer one, that there's 4 roots per length of stem even though there's no phloem. So that is against mass flow. And the last one, there are no leaves, it's just the stem, so there's no photosynthesis to act as a source in this case. Hope you found that useful. Please do subscribe to Dr. Biology, and I will see you soon.